This video will focus on the weight loss pill bitter orange, or Advantra Z, or citrus aurantium. The main proteoalkaloid in this drug is P-Synephrine. P-Synephrine occurs naturally in some plants and animals. It is found in low concentrations in orange juice. Many obesity studies suggest that adaptive thermogenesis is defective, as it is individuals' regulation of healthy metabolism and body weight control, so thermogenic stimulants such as P-Synephrine enhance thermogenesis but are not associated with the central nervous system stimulation that is seen with most thermogenic agents. It does so by binding to beta-adrenergic receptors, increasing the ability of the body to break down and metabolize carbohydrates in skeletal muscle. This is accomplished by facilitating muscle cellular uptake of glucose, glucogenolysis, gluconeogenesis, glycolysis, and oxygen uptake. Glucose metabolism is regulated by glycogen phosphorylase and glycogen synthase. These enzymes are both regulated by phosphorylation. However, phosphorylase is active when phosphorylated, whereas synthase is inactive when phosphorylated. The activation of glycogen phosphorylase and inhibition of glycogen synthase is initiated by epinephrine. Epinephrine binds to and turns on beta-adrenergic receptor in the cell membrane, interacting with the G protein that activates adenylate cyclase. Adenylate cyclase then synthesizes cyclic AMP, or CAMP, from ATP. This CAMP molecule will activate CAMP-dependent protein kinase that will phosphorylate and inhibit kinase, then catalyzes glycogen degradation. P-Synephrine has been found to be an agonist of the beta-3 adrenal receptor, which causes an increase in glycogenolysis. This compound also works to activate initial calcium release. Calcium works with epinephrine to maximize phosphorylase function by binding to and rendering phosphorylase kinase fully active. A study in 2014 by Oliveira et al. Showed P-Synephrine influencing carbohydrate metabolism by mobilizing calcium and upregulating CAMP production. This study used rats and performed liver perfusion by adding P-Synephrine to perfusion liquid. When perfused with substrate-free medium, the liver released glucose, lactate, and pyruvate from glycogen catabolism. This figure shows the changes in glycogen catabolism and oxygen uptake caused by P-Synephrine when 200 micromolars was added at 20 minutes. Glucose release, lactate production, pyruvate production, and oxygen consumption were all measured. All of these were stable before the infusion of P-Synephrine. With P-Synephrine in the system, oxygen uptake and glucose output increased and stayed increased for the entire infusion period. This increase was found to be 0.54 micromolar per minute per gram above basal rates and 120% above basal level, respectively. Lactate production was also stimulated but did not remain stable, and pyruvate production diminished, which can be theoretically contributed to the fact that with increased oxygen levels, pyruvate is converted to acetyl-CoA rather than reduced lactate, and consequently moves through the TCA or citric acid cycle and electron transfer chain, where it can be fully oxidated to ATP energy. Removal of the P-Synephrine from perfusion liquid was accompanied by rapid responses of each factor. This study found that the effects of P-Synephrine on glucose output and oxygen uptake increased to a range of 100 micromolar and remained approximately the same until 500 micromolar. A 2011 study by Stohs et al. Determined the effect of P-Synephrine on resting metabolism, blood pressure, and heart rate. The experiment was a double-blind randomized test that included 50 participants. Prior to the study, participants had their rate of oxygen consumption, blood pressure, and resting heart rate and resting metabolism measured. The compound P-Synephrine was administered through bitter orange extract weight loss pill Advantra Z that contained 50 micrograms of the active ingredient to the experimental group. The control received a placebo. The experiment also studied the effect of P-Synephrine in conjunction with varying levels of hesperidin and naringin, two flavonoids that have been suggested to increase the thermogenic effect of P-Synephrine by enhancing the expression of adiponectin, a protein known to be involved in the regulation of lipid and glucose metabolism. This graph shows the resting metabolic rates expressed as kilocalories in each experimental group and the placebo group after 75 minutes of rest. A small decrease occurred in the placebo group, a result of the continuous fast. Increases occurred in all of the treatment groups. The 50 microgram P-Synephrine group, indicated by group 2, more than doubled their consumption by 65 kilocalories over the placebo. The third group, where participants ingested 600 micrograms of naringin as well as the 50 micrograms of P-Synephrine, further increased consumption over the placebo at threefold, 122 kilocalories. Group 4 was administered 100 milligrams of hesperidin as well as 600 milligrams of naringin and 50 milligrams of P-Synephrine, which showed a further increase in calorie consumption by fivefold, 100 183 kilocalories over the placebo. The fifth and final group, consuming 1,000 micrograms of hesperindin as well as 600 micrograms of naringin, experienced a consumption increase of only 79 kilocalories over the placebo. These data indicate that increasing hesperindin reversed some of the thermogenic effects. The downside of the study is that it is not long-term and therefore could not be used to assess the amount of weight loss due to consumption of the pill. However, FDA reports conclude that there are no adverse side effects associated with bitter orange extract or P-Synephrine.